So we've talked about kind of what life means, and we talked about viruses. And I didn't really give you a time scale of them, but what I want to focus on now is kind of looking at the history of Earth and kind of tying that into life in general. So while I don't mention viruses particularly in this kind of timeline that we're going to go through, they probably were actually the very first things. I hate calling them organisms because they're not. Remember, they're not living, but they kind of co-evolved with life over time. But we're really going to focus on things that are living. So we'll talk about the evolution of bacteria and the evolution of the process of photosynthesis. And we're going to start really at the beginning, the very beginning. And this video is going to go at least to the year 2 billion. So let's go back. Earth is a molten rock of rockiness, and I'm going to call this year 0. So when Earth first formed. And when Earth first formed, this was about 4.5 billion years ago, so quite a lot of zeros in that. To give you some perspective, Homo sapiens as a species didn't really evolve until about 200,000 years ago. So very long time ago that Earth was formed. And in the early stages, Earth wasn't habitable, and it was anoxic. And anoxic means without oxygen. And we'll talk about how that kind of came about and changed over time, but it was an uninhabitable place. We did not have life right off the bat. It was this molten, lots of volcanic gases, and Earth was incredibly hot as well because of this molten nature. Now, as it began cooling, as it began stabilizing, we're going to jump forward in time about 1 billion years. So I'm calling this year 1 billion. The other way you can think of this is 3.5 billion years ago from present. So year 1 billion, we're starting to see the emergence of prokaryotes, particularly from the domains bacteria and archaea. Remember, the 3 domains of life are bacteria, archaea, and eukarya. And so here, this is our prokaryotes, our unicellular prokaryotes. As a reminder, if you are like, oh, prokaryotes, I know that word. So prokaryotes, there are quite a few differences from eukaryotes. The biggest ones, and the ones we're going to explore more in this class, is the lack of a nuclear envelope. So they still have nucleic acids, they still have DNA, but they're just kind of free floating, like you see in this image, kind of free floating within that organism. Whereas in our eukaryotes, that DNA is protected behind a nuclear membrane. The other difference is prokaryotes also lack membrane-bound organelles. They still have a lot of the functions that organelles have. It's just organelles kind of encapsulate those functions in a particular region. Whereas in prokaryotes, they might be happening in different places within that cell, but not necessarily contained within a plasma membrane. Those are the 2 major ones that we'll explore more in this class. And these first organisms, the bacteria and the archaea, the first ones, there's more of them now, were what we refer to as photoheterotrophs. Now, you might be familiar with photoautotrophs. This would be like present-day plants. They are doing photosynthesis, but photosynthesis hasn't evolved at this point. So photoheterotrophs, the photo means light, so they are getting sunlight. But the thing is they have to get already fixed carbon from the environment. And fixed carbon is an organic carbon compound. So I am going to mention photosynthesis as a base point because you're probably familiar with it. With photosynthesis, those producers can take light energy. They can take carbon dioxide. Carbon dioxide is not fixed carbon. Carbon dioxide is inorganic carbon. And that sunlight can break down carbon dioxide in water to reform into an organic or fixed carbon molecule, glucose. But with photoheterotrophs, they don't have the ability to do that yet. They can use sunlight, but they can't take in carbon dioxide. They have to find those sugars, those sugars that fixed carbon, those carbon-hydrogen bonds, they have to find that already existing in the environment, and it does. They essentially have to consume it, and then that light energy can help to break it down. So these organisms, again, are getting energy from light. They can use sunlight. However, they still have to consume stuff. The goal or the purpose of that light is that light does help to break down the things that they consume in order to create the ATP and other energy that they need. You could almost think of this as cellular respiration, but instead of oxygen breaking down the food, it's sunlight breaking down the food. So this is year 1 billion. Now, before I move on, I actually want to focus on bacteria and archaea for just a quick moment. So let's talk about the differences between these two domains. We know they're similar in the fact that they're both prokaryotic organisms, but they do have some differences. So one difference is if you were to look at the plasma membrane, if you think about eukaryotes, you think about human cells, you might be familiar with the phospholipid bilayer. There's a phosphate head and a fatty acid tail. Well, in archaea, their fatty acid tail has these side chains. It's not a beautiful, single and double bonds between carbon. There's some side chains that exist that make these fatty acid tails a little bit more wiry. I don't quite know the best. It doesn't make them nice and symmetrical and nice and parallel. Whereas in bacteria, their phospholipids are more like humans. There are no side chains on the fatty acid tails. The fatty acid tails are fairly parallel to one another. Another difference that exists between bacteria and archaea is the cell wall composition. So something that prokaryotes have is the cell wall. Some eukaryotes have a cell wall, such as fungi and such as plants. Animals lack that cell wall. Now in bacteria, there is a chemical inside of that cell wall called peptidoglycan. Peptido referring to a protein, glycan referring to a sugar. Whereas archaea, although yes, they have a cell wall as well, they're lacking that molecule of peptidoglycan. Instead, their cell wall is made out of a chemical called chitin. And I'll go ahead and spell that on the slide for you because it's not spelled how you think it would be. So archaea in their cell wall have a molecule called chitin. Now I mentioned the molecule it is because I wanted to show you this simplified phylogenetic tree that's showing you the relationships between bacteria, archaea, and eukarya. Now it's very easy to assume that archaea and bacteria are very closely related to one another because they're both prokaryotes. But it's actually quite interesting because scientists believe that if you look at the common ancestor to the domains bacteria, archaea, and eukarya, that there's one common ancestor. Bacteria split off, and then there was another lineage that eventually diverged into archaea and eukarya. And the reason scientists think this is because archaea has actually quite a few similar characteristics with eukarya. One of those characteristics is the fact that archaea has chitin. Chitin is seen in the animal kingdom. Chitin is seen in lobster shells, for example. It's seen, I think of shellfish immediately. It's probably found in other things, but it's seen in quite a few organisms in eukarya. Something else they have in common is the way that the proteins that are used during the cell division and replication process. Archaea employs very similar proteins as eukarya and is actually quite different from bacteria. If you took a cell and molecular biology course, you probably learned a lot about cell division and DNA replication in bacteria. But again, in archaea, it actually mimics more of what eukaryotes do versus what prokaryotes do. So be sure to know this simplified phylogenetic tree. Be sure to understand the relationship between bacteria, archaea, and eukarya in their evolution. Now, there is more differences. We're not really going to explore them. These were just some of the two related to things that you might already be familiar with. Archaea, as the name suggests, are archaic or old. Archaea still exist today. They're found in some more of the extreme environments. Scientists don't know a lot about archaea, and one of the reasons why is it's really hard to mimic the conditions that archaea live in, and so that means growing them in a lab becomes a whole lot harder. We understand some of them, but a vast majority of archaeans, we just really don't understand much. So I'm not really spending that much time on archaea. We're going to focus a lot more on bacteria instead. Before we move on in our future, I'll talk a little bit more about bacteria specifically, so this does not apply to archaea. And I wanted to talk about the two types, or two major groups of bacteria that we see. And these groups are based on the composition of their cell wall. And the reason I mention this is this actually has implications for human health. So that's why I kind of want to throw it in here while we're introducing bacteria. So one grouping of bacteria is called gram-positive bacteria. So take a look at this image that popped up. So the cytoplasm is representing the inside of this bacterial cell. Bacteria, just like eukaryotes, have a plasma membrane. And then they have a cell wall, and their cell wall is made out of a chemical called peptidoglycan. Now, peptidoglycan is, again, this protein sugar thing. The reason this is called gram-positive is that gram is a stain, and this is going to help scientists identify what kind of bacteria they're looking at under the microscope. The gram stain is more of like a pinkish purplish stain, and what happens is it sticks to peptidoglycan. Now this cell wall has tons of peptidoglycan, so this gram stain sticks onto the peptidoglycan, and there's so much peptidoglycan that it stains this really dark purple color. So gram-positive is one style. Gram-negative is actually quite a different style of the way that bacterial cell is assembled, I guess you could use. So again, here's the cytoplasm. This is the inside of the bacterial cell. It's got a plasma membrane. It has a cell wall, but notice how much smaller it is, like literally smaller compared to our gram-positive. This cell wall is still made out of peptidoglycan, but it's a lot thinner. And the other strange or different thing is that this gram-negative bacteria actually has two plasma membranes. So it has a membrane, it has a small cell wall, and it has another membrane. Now, when we stain a gram-negative bacteria, that gram stain again wants to stain onto peptidoglycan, but it's really hard for it to get to it. I mean, that stain has to go through a plasma membrane. And you've probably learned before that plasma membranes are pretty selective. It's going to actually be quite hard for that gram stain to make it through to actually attach to the peptidoglycan. And so because it's not able to stick to it, it gets this lighter color, this light pink color. And again, with gram-positive, there's a whole bunch of peptidoglycan to latch onto, and it's so much high in concentration, it comes off as a purple color. Now, for those of you who have taken micro, you are probably thinking, that's not 100% accurate. And you're right. I am kind of simplifying it because in this class, I don't really care about the mechanism. I do want you to know those color differences, and in general, it's just because of the amount of peptidoglycan that exists. But let's bring this to immune response. Again, keep in mind, plasma membranes, super selective. Now imagine creating an antibiotic, something to kill a bacteria cell. You know, with gram-positive, they only have one plasma membrane they get through. With gram-negative, that antibiotic is going to have to make it through two plasma membranes, and it's going to be a lot harder to kill a gram-negative bacteria. This isn't to say antibiotics can't make it through, but it becomes a lot more difficult to make it through. And this is why it has implications to human health, because it's harder to treat gram-negative bacteria. Not impossible, just harder. One last thing before we continue on with our life cycle of Earth is thinking about antibiotics. I told you that, with gram-negative, because of those two plasma membranes, we can use antibiotics, maybe not as effective, it's not as effective is probably good. So I want to talk about antibiotic use and the idea of antibiotic resistance, which I'll explain a little bit more in a second. But the CDC says it's actually one of the world's most pressing public health problems. Diseases, important. Viruses, bacteria, human health in general, important. But the CDC says that very specifically, antibiotic resistance is going to be the thing that kills us all. But perhaps. So let's talk a little bit about what I mean by antibiotic resistance. Let me get this image up. Is that, like other things we've talked about when talking about evolution, bacteria evolve as well. Now you take an antibiotic, and it might kill 99.9% of the bacteria that it was designed to attack. But bacteria have lots of mutations. We've learned that because of their asexual reproduction and their fast reproductive times, they accumulate mutations pretty quickly. And so that antibiotic might be but so effective, And it's probably going to leave behind some other bacteria, and those bacteria were resistant. Well, they now just lost their competition. So this one is showing the pink ones that are the ones that survive. These purple ones are the ones that died. Well, now these two pink ones are the only ones that survive, are the only ones in this area, meaning they now have a lot more resources. They start dividing and passing on that trait of antibiotic resistance to the next generation, and so on and so forth. And so what's happening is bacteria are essentially just evolving. They're evolving to a selective pressure that we as humans have instilled on them. So every time you take antibiotics, you are contributing to antibiotic resistance. Now, this has become a newer problem, but it was always going to happen. Ever since antibiotics were invented, this has been a problem. But the problem with this problem is that now that more time has passed and more antibiotics have been taken, we're starting to feel the impacts of that. There are lots of antibiotic resistant strains of bacteria out there. And it's terrifying because how do we treat it? Every year in the United States, about 2 million people become infected with bacteria that are resistant to antibiotics. We don't have a way of treating it. We can treat the symptoms. We can make sure you're getting fluids. We can make sure your temperature is not getting too high that you die. But in the end, you're relying on your personal immune system to kill that bacteria. Of those 2 million people, most of them recovered, but still 23,000 people will die from something we can't treat, something we could have used to treat, but we can't anymore. This idea of resistant organisms is not novel, with antibiotics obviously having a huge impact to human health, but this problem has been seen in agriculture. We use pesticides. Well, those pesticides are becoming less and less effective because of evolution. So it's really interesting to kind of apply evolutionary concepts to the health industry and how evolution seems super cool and like you can, see some really amazing adaptations, but it works the other way too. Those adaptations are great for the bacteria, not so great for human health. All right, so we could talk about this forever and we'll talk more about that in class. But again, just wanted to give you some more background about bacteria and its impact to humans before we move on. So let's jump to year 2 billion. So again, year 1 billion, we're starting to see those photoheterotrophs, these organisms that can use light energy to break down organic carbon compounds that they found in the environment. It takes about another billion years in order for photosynthesis to evolve. And the first group of organisms that we see photosynthesis involving in is cyanobacteria. So still bacteria, but this group of cyanobacteria are the ones that can actually do photosynthesis. These are 2 pictures of bacteria that do photosynthesis. And we refer to these organisms as photoautotrophs. They can use light energy just like our photoheterotrophs, but they can use inorganic carbon, like carbon dioxide, in order to make organic carbon, like glucose. They then will do another process to break down that organic carbon into that energy source for them. But the thing is they don't have to consume organic carbon. They don't have to find organic carbon. They can just take advantage of things in the atmosphere in order to absorb that inorganic carbon and then make it into organic carbon. And organic carbon is what is needed by all living organisms in order to break it down to create energy or ATP for the cell. Now, not only was the evolution of photosynthesis kind of, pretty big deal, and organisms being able to make their own compounds, but it made some changes to Earth's atmosphere, changes that are going to kind of kickstart life in other domains. So the first thing that happened was the Great Oxygenation Event. Or I shouldn't say the first thing, but 2 consequences were, 1, the Great Oxygenation Event. The onset of photosynthesis meant that oxygen was a byproduct. Now, while these photoautotrophs would use some of that oxygen, a majority of it was a waste product and was released into the atmosphere. Remember, Earth, up until this point, is fairly anoxic. There might be very trace levels of oxygen, not enough for, say, a human to survive on. But with the advent of photosynthesis, what we start seeing is a lot more oxygen in our atmosphere, going, say, from 2% to 5% to 10%, Currently, just if you're not familiar, currently Earth has about 22% of oxygen in its atmosphere. And so we're starting to go from 2% to 10, upwards to like 12, 13%. So it's increasing a lot. The reason this is important is oxygen is needed for cellular respiration, which is the breaking down of organic carbon to create ATP. Before this, fermentation was pretty much the way it was done. You can ferment without oxygen. And if you remember from way back when, fermentation, while yes, it gets you ATP, it's fairly inefficient. If you're a really small unicellular organism, it's not a big deal. But organisms are limited in size because of the ability to get ATP. So more oxygen means that we can start seeing the evolution of cellular respiration, which means we get more efficiency, meaning we get more ATP, which means we can get bigger organisms. So oxygen, good. And it started with this advent of photosynthesis. The other thing it created was ozone. If you're not familiar, ozone is O3. So when oxygen is emitted into the atmosphere, it's in the form of O2, so diatomic oxygen. In our upper atmosphere, you have a lot of ultraviolet radiation coming in from the sun, and that ultraviolet radiation will split oxygen. And sometimes when it splits oxygen, those free oxygen will find another diatomic oxygen and join it. So again, here's O2. You've got some sunlight coming in. It splits it to two singular oxygens. And some of them will just bind to diatomic to create O3. And O3 is ozone. Now, ozone is an incredibly important molecule. Ozone essentially provides a shield around Earth. Ozone helps to absorb and reflect the most severe of the ultraviolet radiation that comes in from sun. If you're familiar with the rays, it pretty much blocks out exclusively or 100% UVC. UVC is the most potent and most mutation-causing form of ultraviolet radiation. It's the strongest form, so to speak. And so ozone acts as a shield, which is great for organisms on Earth. At this moment, organisms and bacteria pretty much live exclusively in water. They really can't survive on land. Sometimes they need an aqueous solution, but the other problem too is that ultraviolet radiation causes mutations in their DNA, typically mutations that cause them to die. So life is pretty much exclusively stuck to the oceans. But with the advent of ozone and the creation of the ozone layer, which is going to take quite a while to build, but the creation of that layer is also making it that land on Earth is going to become a more habitable place to live. Whereas beforehand, it wasn't. That radiation was so high, it would be impossible to survive there. So that's where we're going to stop with our timeline for now. So again, it was year 1 billion that we saw our first organisms, our prokaryotic bacteria and archaea. They were able to use and take advantage of light energy, but they still had to consume organic compounds in order to create ATP. Whereas about 1 billion years later, in year 2 billion, we start seeing the advent of photosynthesis, which increased oxygen in our atmosphere and also increased ozone in our atmosphere, leading the way for evolution later in life.